Assalamu alaikum warahmatullahi wabarakatuh. In our video, we will discuss the method and test for evaluation of a semi-solid dosage form. First of all, the semi-solid dosage form, such as ointment and cream, are dermatological preparation intended to be applied externally on the skin, either to be used topical or transdermal. So when used as topical, we will apply it to body surface such as the skin or mucous membrane, for example, nasally, vaginally, or rectally, to treat some disease. So about using it as a transdermal, the drug will deliver through the skin to general circulation for systemic effect. This system follows absorption of an active ingredient through the transdermal barrier. Therefore, they may use it for a therapeutic, protective, or for a cosmetic function. So before talking about the method for an evaluation, we must know what's the ointment, which are drug substance that dissolve it or disperse it or emulsify it in a suitable ointment base, either to be used for medicated or non-medicated purpose. Therefore, any manufacturer will search about the ideal properties of an ointment. Even the patient will search for that. So it must be first pharmaceutically elegant and has low sensitization index, low index of irritation to avoid the harmful effect, and must be compatible with the skin, also physically and chemically stable, because the stability will affect the efficacy and the safety of a dosage form. Another properties will affect the dosage form is a high degree of consistency and the spreadability, and being non-greasy, non-dehydrating, and non-staining. This factor is important because will affect the competence of a dosage form. Finally, the release of an active pharmaceutically ingredient efficiently at the site of an application is an important factor for producing sufficient therapeutic effect at the site of an application. These factors or properties will give the ideal form of an ointment. So there is some test to evaluate the ointment. The importance of applying this test for every ointment to ensure the efficacy and the safety of the physical and the chemical properties of all ingredients present. To be able to release this ointment to the market and having pharmaceutical evidence to get the FDA license. This test is taken from the pharmacopoeia, which are test of a rate of absorption, test of a non-irritancy, also test of a rate of penetration, test of a rate of a drug release, in addition to test of a rheological properties, test of a content uniformity, also test of a preservatives efficacy, and lastly, test of a sterility.
Now I will talk about each test. First of all, test of rate of absorption. This test measures the rate of drug in blood stream. It can be done in vivo only. The absorption test will be done by rubbing the ointment over a specific area of the skin. Then we should analyze serum and urine sample to measure the quantity of drug absorbed. Next one is test of non-irritancy. It's evaluated by patch test. In this test, we need 24 human volunteers. The ointment is applied on the back or arm for 21 days, and we will observe if the skin has any sensitivity or visible reaction. So after that, test of rate of penetration. In this test, we applied quantity of the preparation over a specific area of the skin at a period of time. Then the preparation left over is collected and weighed. Then we measure the difference between initial and final weight to evaluate the rate of penetration. This test should be repeated more than once. Next one is test of rate of drug release. It's performed by applying small amount of ointment on the surface of nutrient agar. If the drug is bactericidal, the agar plate is seeded with suitable organisms. After a period of incubation, the zone of inhibition is measured and correlated with rate of release. After that, test of rheological properties to evaluate the viscosity of the ointment by using cone and plate viscometer. Next one is test of content uniformity. Determine the net weight of 10 ointment container. The result should match each other and with labeled quantity. After that, test of preservative efficacy. It's performed by using Bohr technique. Determine number of initial microorganisms. The solution of different samples are mixed with tryptone azolectine. Then add the culture of microorganism into each mixture. All mixtures are incubated. The number of microorganisms in each sample will be counted on 7th, 14th, 21st, and 28th days of inoculation. On 14th day, the number of vegetative cells shouldn't be more than 0.1% of initial concentration. On 28th day, the number of organisms should be below or equal to initial concentration. Final test is test of sterility. It's for detection of microbial contamination in ointment. The method involved dissolving the ointment in filter sterilized isopropyl myristate and filtering through a membrane filter. So, most of the semi-solid preparation as ointment and cream are applied to the skin or mucous membranes such as rectal, urethral, vaginal, nasal mucosa, and cornea. So, semi-solid preparation must be evaluated and tested. To avoid any problems such as irritancy, poor absorption, or penetration, and to ensure safety and quality of all material. Quality control personnel should be applied each test to get high quality and efficiency for each semi-solid preparation.
Another type of semi-solid is a cream, which is contain one or more medicinal agent divided into two types, oil in water or water in oil. It appears like a low viscosity and creamy white. Requirement of a cream: it should liquefy at body temperature and its viscosity should be low enough to spreading and high enough to retain the suspension particle. It should be emulsion type with a small percentage of water and penetrate the epidermis. We have 4 tests to evaluate the cream: spreadability, rheology, sensitivity, minimum fill. Minimum fill: this test compares the weight of a product, mainly used for content uniformity of a product. Procedure: first step is weight of a product and its container. Then weight the empty container only. The difference between these two values give the weight of a product. If the average weight of 10 container is less than 60 grams, the label amount should not be less than 90%. And if the weight between 60 and 150 grams, the label amount should not be less than 95%. Also, this test not used if the average weight more than 150.
It is obvious that the efficacy of topical therapy depends on the patient spreading the drug formulation in an even layer to administer a standard dose. Therefore, spreadability is an important characteristic of semisolid, and it is responsible for correct dosage transfer to the target site. There are many factors that can affect spreadability. Uh, First is the formulation characteristics. This include viscosity, elasticity, and rheology. Second, the rate and time of shear. Also, the temperature plays an important role, as well as the site of application. There are many methods to measure spreadability, such as parallel plate method, the subjective assessment, and the master curve method. And we will talk in details about each one of these methods in the next slide. First is the parallel plate method. This method is the most widely used method for determining and quantifying the spreadability of semi-solid preparations. The advantages of the method are simplicity and the relative lack of expanse. It can be determined by sliding a glass plate over the product, in which the product is placed on another glass plate fixed on a wooden block. Then weights are added to the pan so that sliding of the movable glass plate is obtained. Second is the subjective assessment method. As the name suggests, the method is based on the sensory assessment of a human volunteers. So, the measurement is closest to the true spreadability. In fact, it is reflective of the true spreadability because the results are generated from individual feel of the formulation. Since the formulation could be spread on the backs of the hand. The scores obtained will be plotted against the viscosity of the sample. Some scientists stated that subjective spreadability and viscosity are perceived as the shear stress on the fingers, whereas subjective thickness of formulation is perceived as the time frame during which the finger is pulled away from the sticky surface. Third method is the master curve method. This method combines both the advantages of subjective assessment of spreadability and the use of instrumental measurements. This method determined the relationship between the shear stress and the shear rate that operated during the spreading of topical preparation on the skin. A panel of volunteers compared a series of formulation that varied in consistency from mobile liquids to stiff semisolids with a range of Newtonian silicone oils of various viscosities. Third test is rheology test. Rheology is very important as these creams are marketed in tubes or containers. The rheology or viscosity should remain constant, as these products are normally non-Newtonian in nature. Viscosity can be measured using viscometers used for such liquids. Rheological measurements are utilized to characterize the ease of pouring from a bottle, squeezing from a tube or other container, maintaining product shape in jar or after extrusion, and rubbing the product onto and into the skin, and pumping the product from mixing and storage to filling experiment. Next is sensitivity test. As various types of ingredients are used with occasional use of antiseptics, hormones, etc., there is a possibility of sensitization or photosensitization of the skin. This should be tested beforehand. This test is normally done by patch test on and can be either open or occlusive. The test sample is applied along with a standard market product at different places, and effect is compared after a period of time. In this video, we covered the most important tests used in the quality control process of semi-solid dosage form. I hope you find it clear and informative, and thank you for watching.